with biochemistry. You can see biochemistry is divided into molecular, cellular, laboratory techniques, genetics, nutrition, metabolism. The first one we discuss now is molecular part. In the molecular part, of course, we need to start with the DNA. DNA is very large molecule. We need to fit the DNA into small nucleus. So first DNA mixes with the proteins and forms chromatin. Next it fuses with the histone to form nucleosome. So you can see these are the nucleosomes. Next, especially the H1 histone will bind with them so that it will form a very condensed structure. And remember, histones are positively charged, DNA is negatively charged. So H1 histone will also attach it with the linker DNA. Okay, so histone is very large. So how to remember that? With the mnemonic of amino acid, it's lysine and arginine. Lysine L, AR is for arginine. And this DNA and histone both synthesize in the S phase during mitosis. And mitochondria, mitochondria also have DNA, but that is circular DNA. It is not like a double helix DNA. It is a circular DNA, and it does not utilize any histone because it is directly circular. But here it is double helix, so it is first converting into chromatin. Next to the nucleosome, it is first converting to chromatin with the help of protein. Next to the nucleosome with the help of histone. Of course, histone is also a protein. Next, it is converting into supercoil structure because of H1 histone protein. So after all this process, finally it will convert into heterochromatin and euchromatin. We need to understand difference between hetero and euchromatin. In the name itself, you can see euchromatin. Euchromatin means it is ready for the transcription. Heterochromatin is not ready for the transcription. It is a it appears darker. You can see here in this diagram, this darker one is a heterochromatin. H here it is represented. It is inaccessible, that is it is inactive, transcriptionally inactive. It will not transcribe, so that it can't produce any proteins. The reason is there is more methylation here. Actually, remember, whenever there is methyl group added to the DNA, it will lead to mutate. It will mute the DNA. So how to remember that? Methylation, M for methylation, M for muting of DNA. Okay, so that is numeric. Of course, acetylation will lead to activation. A for A. Acetylation will lead to activation of DNA. So here the activation should drop, that is acetylation should drop, and the muting should increase, that is methylation should increase in heterochromatin. Okay, so this heterochromatin examples are like bar bodies which are inactive X chromosome. It is visible on periphery of nucleus. Next, coming to euchromatin, and the name itself, you can see they are transcriptionally active. So now you can easily guess there is low methylation in euchromatin and there is more acetylation, because acetylation means more active in transcription. Okay, now coming to DNA methylation, I already said you now that methylation is nothing but adding of methyl groups to the DNA and making it mute. So actually bacteria DNA will undergo methylation process, especially in cytosine and adenine amino acids, because they want to protect themselves from the viruses, especially bacteriophages. They want to protect themselves from bacteriophages. So bacterial DNA will undergo methylation. In humans, 70% of our DNA is methylated. That is, it is muted, or sometimes muted. Especially here also in humans also, especially they attack on cytosine and adenine amino acids. Next, coming to histone methylation. So here also, if histone is methylated, indirectly transcription is stopped, right? Because histone is only coiling the DNA into smaller part. So histone methylation also muting. But important is to know the amino acid, that is lysine and arginine. How to remember this? Histones are large, so lysine, arginine. Next, histone acetylation. I said you already, acetylation means activation. Here also acetyl group is added to the lysine, so more activation means more transcription. Histone deacetylation means reverse of acetylation, that is inactivation. So this is also leading to decrease in transcription. Here it is anti-cancer. Can we use this anti-cancer? Because there is low transcription, means low protein formation. So it is can be used as anti-cancer. And histone is also responsible, this deacetylation is responsible for Huntington disease. This is a neuronal cell death which is seen in striatum. That disease is associated with histone deacetylation. So these are the main high yield topics here. First you need to know about chromatin structure. Next you need to know about DNA methylation. Next histone methylation, histone acetylation, histone deacetylation, of course heterochromatin, euchromatin. So this page have this all important points. And one more thing is drug induced lupus. By taking some drugs you may get lupus, that is drug induced lupus. Actually what you see in drug induced lupus, you get fever, you get some rash and joint pain. So these are the features: fever, rash, joint pain. After using some drug, if you get these features, then you should suspect drug-induced lupus. Here, the main problem is anti-histone antibodies. That's why we are discussing here. Anti-histone antibodies means antibodies attacking on histone. So, because of that, there's a problem in supercoiling of DNA, and that is leading to this problem. So, what drugs are causing this? Hydralazine, procainamide, isoniazid. Isoniazid, tuberculosis drug, procainamide, anti-arthritic drug, and hydralazine. So, all these three drugs are causing drug-induced lupus. Next, coming to nucleotides. So nucleotides and nucleosides, we need to know difference between this. How to remember this is nucleotide, tide of waves, it has some phosphate, okay, and nucleoside only have sugar base. So side, sugar, base, okay, only base and sugar. Base is here nitrogenous base. Nucleotide also have nitrogenous base, sugar, but phosphate is extra for here, tide, phosphate T. So this is the mnemonic. And one more thing, if phosphate is in mono form, it will not attach to DNA. If phosphate is in tri form, triphosphate, then only it can be attached to the DNA. And next, coming to purines and pyrimidines. Actually, what are these? These are both the nitrogenous bases. So these nitrogenous bases are common for both. You can see here nitrogenous bases. These are common for both our nucleoside and nucleotide. In both these bases are present. So purines, the mnemonic is as pure as gold, because purines as pure as gold. As means adenine, G for guanine. So these both are present in purines. Pyrimidines, you have cut. Mnemonic is cut. That is, you have cytosine, uracil, and thymidine, and thymine, uracil, thymine. So one thing you need to remember, thymine is present only in DNA, uracil is found only in RNA. So these are all the basics things we all know. But the important deamination reactions is cytosine can convert into uracil when undergoes deamination. Same way adenine to hypoxanthine. This we'll discuss in purine synthesis pathway. Guanine to xanthine, 5-methyl cytosine to thymine. So these are some important deamination reactions. So next thing is we know that this cytosine will attach to guanine. These are called base pairing. This base pairing, that is nitrogenous base, undergo pairing. Between C and G there is triple bond. Between A and T there is double bond. So always whenever the DNA want to replicate, it want to break this A and T bond, because there are only two bonds. It can easily break this. Actually, this process is under DNA helicase enzyme. We'll discuss it in next page. So A double bond T, C triple bond G is called as base pairing. Very important topic. And one more thing, increase Cg content, increase melting temperature of DNA. This is also important. And linked by 3 to 5 phosphodiester, the backbone of DNA, or you can say this nucleotides, nucleosides, are linked by mainly 3 to 5 phosphodiester bond. Now pyrimidine and purine synthesis. So first let us begin with purine synthesis. So for purine synthesis you need sugar, that is ribose 5-phosphate. So ribose 5-phosphate, which is coming from HMP shunt, the first thing you need. Next you need some amino acids. The mnemonic is GAG, that is you need glutamate, aspartate, glycine. Of course along with this you need carbon dioxide, tetrahydrofolate. So these are the ingredients which you need for the purine synthesis. Now let's begin the process of purine synthesis. The first enzyme you need is PRPP. PRPP is phosphoribosyl pyrophosphatidyl synthase. So you can remember in short form as PRPP. So first ribose 5-phosphate in the presence of PRPP will convert into IMP, and this IMP will convert into AMP and GMP. That's it. This is the purine synthesis. Very simple. First you have ribose 5-phosphate, next you have PRPP, next you have IMP, next you have AMP, GMP. And one more thing, from IMP to GMP you need one enzyme, that is IMP dehydrogenase. Inositol monophosphate dehydrogenase is required for conversion of IMP to GMP. So this is purine synthesis. We need to see the purine salvage pathway, that is how again we can get this GMP, IMP and AMP. We can get it again. How, let us see. So again the same ribose 5-phosphate PRPP synthase. It will form, IMP will form into GMP, AMP. For IMP to GMP you need IMP dehydrogenase. Okay, now they are formed. Now what happens to IMP is it will further convert to inosine, and this inosine will convert into hypoxanthine. From hypoxanthine, in the presence of xanthine oxide, xanthine oxidase, it will convert into xanthine. Again, by presence of xanthine oxidase, it will convert into uric acid. Actually, it excretes in the form of uric acid. Again, in the presence of urate oxidase, it will convert the uric acid into allantoin. So this is one pathway from IMP to inosine, from inosine to hypoxanthine, hypoxanthine to xanthine, xanthine to uric acid to allantoin. Next, there is another pathway. IMP will first convert into GMP. Next, AMP. Now GMP, what will it undergo is it will convert into guanosine. Finally, it will form guanine. Next, coming to AMP. AMP will convert in the presence, not in the presence of APRT. AMP will convert into adenine. That's it. Very simple. GMP to guanine, AMP to adenine, but IMP first to inosine. Next, after inosine to hypoxanthine, hypoxanthine to xanthine, from xanthine to uric acid to allantoin. So now it undergoes a salvage pathway. Salvage pathways in the reverse. Hypoxanthine, guanine, both in the presence of HGPRT and PRPP, they can convert again back into IMP and GMP. And same, adenine in the presence of APRT, PRPP, it can again convert back into AMP. And AMP can again form adenosine. And adenosine in the presence of ADA, that is adenosine deaminase, it can convert into again inosine. So that this is like salvage pathway coming back, and again it can form all the components. So adenosine again can form inosine, and we continue this pathway. So like this, all these pathways are united. So now what happens is there are some defects. Like for example, if you take here ADA, if ADA is not present, that is adenosine deaminase deficiency, you can understand adenosine will not convert into inosine, and this is ADA is required. So that will lead to one of the major causes of autosomal recessive SCID. You can see I mentioned SCID. It is also autosomal recessive problem. They don't have ADA, so they can't convert this adenosine into inosine. So because of that, that will lead to decreased ribonucleotide reductase activity. That will lead to decreased DNA precursors in the cell, so that the lymphocytes in the cell will decrease. You can see here, decrease ADA, increase DATP, increase DATP, decrease ADA. Because of this process, of course, ADA is decreased here, that is adenosine deaminase. Increase DATP. DATP is the one which increases because of this. So this is adenosine deaminase deficiency. Next is very important syndrome, that is a Lesch-Nyhan syndrome. Very important syndrome. Here what happens is, you can see here HGPRT is absent in it. So if HGPRT is absent, you can understand hypoxanthine, guanine, they can't convert back again into IMP and GMP. So because of that, there is a problem like the PRPP will undergo more activation, and it will lead to formation of more uric acid because of absence of this HGPRT. So more uric acid means you can understand uric acid accumulation leading to different symptoms. The mnemonic for this remembrance of Lesch-Nyhan syndrome symptoms are HGPRT itself. Here H stands for hyperuricemia. I have explained, because more uric acid because of no presence of HGPRT. G for gout because of more uric acid. P for pissed off. Pissed off means there is more aggression in this patient and more self-mutilation. And R for red or orange crystals. Because of more uric acid, they are now forming crystals of red or orange color. And there are tense muscle because of more uric acid accumulation, that is dystonia. So all this is happening. So now treatment for this is allopurinol or febuxostat. What they do is they just simply inhibit this xanthine oxidase. If you inhibit the xanthine oxidase, you can't produce more uric acid. So that is the treatment for gout and also this Lesch-Nyhan syndrome, allopurinol, febuxostat. And one more thing, this Lesch-Nyhan syndrome is more common in male who are having X-linked disorder. This is a X-linked disorder, most common in male. So this is about purine synthesis, and in purine synthesis there are some other drugs, like you can see here 6-mercaptopurine, azathioprine. These are the inhibitors. These are inhibitors of conversion of PRPP to IMP. So there will be less IMP, more PRPP because of this inhibition. So actually these are pro drug which inhibit purine synthesis. Uh, Next, you have another drug like mycophenolate and ribavirin. Ribavirin is antiviral. Mycophenolate is immunosuppressant. Actually, both of these drugs inhibit IMP dehydrogenase so that the GMP can't form. So these are the drugs you need to remember: 6-mercaptopurine, azathioprine, mycophenolate, which is immunosuppression, ribavirin, which is antiviral. So we are done with purine synthesis. Now let us come to pyrimidine synthesis. For pyrimidine synthesis, first you need amino acids. So remember, purine are, as I said, as pure as gold, mnemonic, right? As pure as gold, and they're also very sweet because they start with sweet, ribose 5-phosphate. Here coming to cut, that is our pyrimidines. So they start with amino acids and carbon dioxide, glutamine plus carbon dioxide both together in the presence of carbamyl phosphate synthase 2. Because of this enzyme, this glutamate and carbon dioxide, they form the carbamoyl phosphate, and this carbamoyl phosphate in the presence of again aspartate. So this pyrimidine synthesis is more using amino acids. First glutamate plus carbon dioxide, in the presence of carbamyl phosphate synthase 2, it is forming carbamyl phosphate. Now again in the presence of aspartate, it is forming orotic acid. This is very important. And orotic acid along with PRPP and UMP synthase, it is forming UMP. So we need to see one thing about UMP synthesis. It is impaired in orotic aciduria. So when UMP synthesis is not present, orotic acid will accumulate in the body, and that is called orotic aciduria. Actually, this is a autosomal recessive disorder in which there is megaloblastic anemia, and the treatment is uridine for orotic aciduria. Okay, now this UMP synthase will convert orotic acid to UMP, and this UMP will further divide into CMP and TMP, but the main thing is UMP converting to UDP. You can just don't remember them, it's okay, but if you remember, it's okay. UMP to UDP, just convert the M to D, that's it. UMP to UDP. UDP again in the presence of ribonucleotide reductase, it can form DUDP. Just add D here because it is ribonucleotide reductase, so it is reduced, so DUDP. And DUDP will convert into DUMP. And dump in the presence of thymidylate synthase will form DTMP. And actually here the some process is going on, like dihydrofolate is in the presence of dihydrofolate reductase, it will convert into tetrahydrofolate. Dihydrofolate in the presence of dihydrofolate reductase convert into tetrahydrofolate, and tetrahydrofolate can again further divide into N5 and 10 methylene tetrahydrofolate. So this process goes on here. So DUMP to DTMP, the final product is DTMP in our pyrimidine synthesis. Okay, now we will also see the drugs which are involved here. So once again, if you want, I'll revise it. Glutamate plus carbon dioxide in the presence of carbamyl phosphate 2, it will form carbamyl phosphate. Carbamyl phosphate with the presence of aspartate, with the presence of aspartate, it will form orotic acid. Orotic acid with PRPP and UMP synthase will form UMP. UMP to UDP, UDP to DUDP to DUMP, DUMP to DTMP. This is the process. Now let us see the drugs. The first one is leflunomide, which inhibits this conversion of carbamyl phosphate to orotic aciduria. And next you have another drug, that is here methotrexate, TMP, that is trimethoprim, and pyrimethamine, and also sulfonamide antibiotics. All this inhibit this dihydrofolate reductase so that tetrahydrofolate will not form. If tetrahydrofolate will not form, DUMP can not convert into DTMP, so that DTMP will be decreased here. And same for 5-fluorouracil and capecitabine. If they inhibit thymidylate synthase, then DTMP can't form. So by this cycle, if you understand, then many drugs, you can see here various immunosuppressants, anti-neoplastic, antibiotic drugs are functioning by interfering with this nucleotide synthesis. So that's why we are learning this cycle, to know about all those drugs which are involved here. And one more important thing which you need to see here is this cycle. If you remember this dihydrofolate to tetrahydrofolate. Actually here in the name itself you can see folate. So whenever there is folate deficiency, DTMP will be decreased. That is very important. So folate is required. Also vitamin B12 is required for conversion of N5 N10 this tetrahydrofolate to normal tetrahydrofolate. You need vitamin B12 and also folate. So whenever this vitamin B12 and folate are dropped, there will be increase in homocysteine. Here you can see clearly homocysteine, actually it is converted into methionine in the presence of vitamin B12 and folate. So when folate and vitamin B12 both are dropped, then that will lead to increased homocysteine. And next one more thing is methyl malanoic acid is converted into methmalone CoA. Methmalone CoA in the presence of B12 converted into succinyl CoA. So if there is no vitamin B12, that is in vitamin B12 deficiency, there will be more methyl malanoic CoA. That is important. So these are both important things related to B12 and folate. Now let us look at some genetic code features. Actually the genetic codes, for example, you take AAU, example, is just example. So if you take this genetic code, this codon only specifies for one amino acid, like that. And next, degenerate or redundant, that is some of the amino acids may also have multiple codons like AUA or GAG, example. This both code for only one amino acid. Sometimes even like that it may be. That is called as wobble. Wobble means there is there are multiple codons for one amino acid, but wobble is that even if you change, for example, GAG, AUG, this both are same codon for only one amino acid. That is very important. This is called as wobble, but it is not for everyone. For example, you can see methionine only have AUG, tryptophan only have UGG. Only they are coded by one codon. And next, common non-overlapping. Like you should read the codon from a fixed starting point, and it is continuous sequence of bases. Next, universal. This genetic code is same conserved throughout the evolution. Exception is for human mitochondria, and here commonless non-overlapping is exception for some viruses. Now let's begin to understand DNA replication. So actually, if you observe the DNA, they are in an anti-parallel manner. That is, if you see one strand, it is from 5' to 3' end. So let me mention here, this is 3' end here, and this is 5' end. Okay, so if you see 5' to 3' end is one strand of DNA, other strand of DNA is from 3' to 5' end. Okay, you can see. So this is called anti-parallel manner of arrangement. Always remember that the DNA replication, actually this is the old DNA, this is a new DNA, now it is forming. So the always a new DNA will start from 5' end to 3' end. It will always undergo synthesis from 5' to 3', and this is a continuous process in the case of leading strand. Actually DNA have two types of strand. So first, if the DNA is separated, the upper strand is called leading strand and the lower strand is called lagging strand. So leading strand, you can say, discontinuous, and lagging strand is discontinuous. Lagging strand is discontinuous because it's having multiple primers, so that it is discontinuous, and this is called as Okazaki fragments. Okay, and one more thing, the DNA replication is semi-conservative. Semi-conservative means one strand is old strand and newly forming strand is new one. So one new one old one, that is semi-conservative. And next one, this DNA replication is more complicated in eukaryotes when compared with prokaryotes. Okay, and first we'll begin with this origin of replication. You can see this is the origin of replication, that is from the 5' end, it continues to the 3' end. Here it is very much seen, this origin of replication is seen in A double bond T because it has only two bonds, but in C triple bond G you can see triple bonds, and it also starts in the Tata box like promoter regions. Actually there are some promoter regions which are present. You can see this yellow color represents promoter region. So this is called origin of replication. Next, coming to replication fork, this is called as replication fork. Here you can see this Y shaped one is called replication fork. This is along the DNA template where the leading and lagging strands are synthesized. You can see this is the leading strand and this is the lagging strand, and this area, Y shape area, this is called as replication fork. Next, let us see helicase. You can see this blue color protein. This is helicase protein, actually, which unwinds the DNA template at replication fork. You can see it is unwinding it. It is dividing into two strands by helicase. Helicase, you can remember, it halves the DNA. But remember, there is one important syndrome called Bloom syndrome. In Bloom's syndrome, there is deficient of helicase, so that the DNA can't unwind like this because of BLM gene mutation. Bloom BLM gene mutation. Now coming to single stranded binding proteins. You can see there are some single stranded binding proteins. These are the proteins which stabilize the single strand of the DNA, so that again they will not coil together and there will be no they'll be stabilized and they'll not coil or re-anneal again. Next, coming to DNA topoisomerases. So in this diagram you can see topoisomerases. Actually topoisomerases are two types, topoisomerase 1, topoisomerase 2. Actually what they do is they remove some supercoils which are present in the DNA, they unwind them. So sometimes also they are needed if they are overwinded, so they will cut them if they are overwinded. If they are if they are supercoils, then they'll try to remove them. So actually this kind of thing are this topoisomerases are present in both eukaryotes and prokaryotes. In eukaryotes they are called, as you can see, irinotecan, topotecan, tecan means they inhibit topoisomerase. So something which is inhibiting topoisomerase means they don't want to cut or repair the uh, DNA, so that will lead to some mutations or problems. And etoposide, teniposide, these are also inhibiting, especially topoisomerase 2. Etopo, tenipo, 2. Topo, irinitotecan, topoisomerase 1. Next, in prokaryotes, you have fluoroquinolones which inhibit topoisomerase 2, DNA gyrase, and topoisomerase 4. Now coming to primase, another one, that is primase. You can see here this is a primase which makes an RNA primer. Actually for the starting of this DNA replication process, first the RNA primer is required. It will convert into DNA by especially DNA polymerase 3 enzyme. DNA polymerase 3 enzyme requires RNA as primer, and next it will convert into DNA and it will start the replication process. Now let us discuss about important protein DNA polymerase 3. Actually it elongates the leading strand by adding deoxynucleotides to the 3' end. It starts adding to the 3' end a new DNA and elongates the lagging strand until it reaches the primer of preceding fragment. For example, you can see here it will start and until again new primer forms. Till then it will form like a bridge like structure, that is DNA polymerase 3. It is most important in prokaryotes. Next, coming to DNA polymerase 1, you can see here this is also same like polymerase 3, but it is also present only in prokaryotes. It degrades the RNA primer and replace it with DNA. So DNA polymerase 3 will only form a bridge like structure, but polymerase 1 will replace the RNA to DNA. Very important function of polymerase 1. Next, coming to DNA ligase, according to the name you can see it will ligate the parts which are like broken, so they catalyze formation of phosphodiester bond within a strand of double-stranded DNA. Especially they join the Okazaki fragments in the lagging strand. You can see in the lagging strand these Okazaki fragments are ligated by means of DNA ligase. Next you have telomerase. So actually telomerase is nothing but, I will draw here, for example this is a DNA strand, you add a tail like structure. Adding a tail is nothing but telomerase. That tail, that too, it has a TTA triple G tag. So you are adding a tag, that is a telomerase function. Why you add a tag is because, remember, especially you add a tag only to the 3' end. You add tag to the 3' end. Why you add is because by multiple divisions of a cell, what happens is some of the DNA pieces are lost, so that we add a new tag so that the cell will remain always young and active. So this tag is actually lost in the case of aging, progeria, but it is, this tagging is increased in the case of cancer, because cancer cells try to prevent from breaking their DNA, so they tag it more so that they can live for a long time. So this is all about DNA replication. In the next video we'll discuss about DNA repair.